Assalamualaikum warahmatullahi wabarakatuh. My name is Dr. Mansyah with my partner Firjatullah Farnas Afalsani. We are from team Hipotesa 1 Jembrana. We are going to present our research entitled Marine Biopesticide Made from Diadema cytosum Control Aedes aegypti larva.
The background of this research is there are many populations of sea arsen type Diadema cytosum which is rarely utilized by people. Sea arsen is rarely utilized because it contains a dangerous poison. The utilization of it is limited. People only take its egg as food, while its shell and spine are thrown away. There is a useful poison found in its shell and spine which can be utilized as biopesticide. Therefore, it can reduce the use of chemical pesticide. The use of chemical pesticide has a side effect, which is it may kill non-target organisms and make the soil dry. In addition, dengue fever is caused by dengue virus, which is brought by Aedes aegypti mosquito. So, based on the background, our formulation of problem is how is the potential of poison found in sea arsen type Diadema cytosum as a biopesticide in controlling the life cycle of Aedes aegypti. The research objective. There are two objectives in this research. The first one, to know the potential of poison in sea arsen type Diadema cytosum as a biopesticide in controlling the life cycle of Aedes aegypti. The second, to produce biopesticide in controlling mosquito larvae. Research methodology. This research was conducted in June to August 2020 at Biology Laboratory of MAN 1 Jembrana. Next, the presentation will be continued by my friend.
Next, there are 5 steps in this research. The first one, we prepare the sea urchin. The second one, we extract it. The third one, production of biopesticide. The fourth one, we test toxicity of it. And then last one, we analyze data. In preparing step, there are 4 steps. The first one, we research to sample sea urchin at Gilman Bay. In a sanitation step, we meat is taken out and its shell and spine are cleaned. After that, it's dried under the sun. And then it is pounded and then is filtered until it becomes Simplicia. The next step, extraction. The fine Simplicia of shell is measured with 96% ethanol for 3 times for every 24 hours in ratio of Simplicial and solvent 1 to 3. The result of maceration is distilled. Then it is evaporated until we get the ethanol fraction. Ethanol fraction is partitioned with N-hexane, so that we get the N-hexane fraction. Then it is partitioned with ethyl acetate. Every solution passed is taken to be tested as a biopesticide. The next step, providing Aedes aegypti. Aedes aegypti egg supply were obtained from Entomology Laboratory of Veterinary Faculty at Institut Pertanian Bogor, West Java, Indonesia. The hatching process was conducted at Biology Laboratory MAN 1 Jembrana. The application of sea urchin poison extract as biopesticide. We make of main solution and dilution. The first, making the main solvent with concentrate 2000 ppm, diluting biopesticide with concentrate 80, 110, 140, 170, and 200 ppm. Using positive control with Abate and negative one with Aquades. Repeat 3 times for each steps. The next step, we test toxicity of it. We provide 3 cups for each concentration. Each concentration is given 10 seconds in star Aedes aegypti larva. This observation was done for 24 to 48 hours. You can see on the picture, it is second star Aedes aegypti larva. It was dark black chiffon. It is Aedes aegypti that graphic for 24 hours treatment. It showed ethyl acetate compound can kill a hive mosquito larva with concentrate 140 ppm. And this is Aedes aegypti that graphic for 48 hours treatment. It showed ethyl acetate compound can kill all mosquito larvae with concentrate under up to 140 ppm. The result of positive and negative control. Based on table, it indicates that our result is more effective than positive control by using Abate. We move result and discussion. The result of this research indicate that the ethyl acetate fraction sample has the highest activity compared with two other examples, N-hexane and ethanol. It can be seen from the high death rate in 24 to 48 hours on the mosquito larvae which have been used for the object of experiment. Sea urchin poison extract can kill a half of mosquito larvae with concentrate up to 100 ppm. It showed the level of toxicity from the poison itself. The calculation of LC50 shows that the ethyl acetate fraction is the most synergistic in controlling the survival of Aedes aegypti larvae with a concentrate of 28.98 ppm. A solution can be said to be very toxic if it has a value less than 30 ppm. The result of this research is supported and strengthened with ANOVA test from SPSS 22. It showed the person is significant to control life cycle of Aedes aegypti larvae with 95% reliability. So, it is supported by the result of the comparison of mosquito larvae death rate on positive control. It indicates that the poison extract is more effective in controlling the life cycle on the larva. The result of ANOVA test of SPSS 22 in 24 hours. There are three table ANOVA test result of SPSS 22 program on 24 hours observation. You can see on the circle it is significant result. In conclusion, the diademasitoxum poison extract is effective to be a biopesticide to control the life cycle Aedes aegypti larvae with concentrate 28.98 ppm ethyl acetate fraction. It is significant to control the life cycle of Aedes aegypti larvae based on the ANOVA test program of SPSS 22 with 95% of reliability.
That's all from us. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.